Hi guys, so today we'll be talking about the HIV, human immunodeficiency virus. That is going to be our topic for today. So what is this whole concept of HIV? Let's write it down here. All right, let's write it down here. What is HIV? Remember, this is called human immunodeficiency virus, right? And I actually drew this diagram of like exactly how this HIV looks like. But before we go over there, let's talk about what is this HIV or the family that it belongs to. We'll talk about this, all right? So HIV is basically remember, it is a positive sense. It's a single-stranded RNA virus, all right? That's what this HIV is. And this is an enveloped virus, all right? This is what you have to remember. It's an enveloped virus, all right? Because why it's enveloped? Because it takes the lipids from its host, right? So it's envelope virus. And this envelope virus, what did it contain? It basically have this capsid, right? It has this conical shaped capsid, which is basically called icosahedral, all right? And inside the icosahedral, it basically contains two identical genomes. We'll talk more about this. But one thing that you have to remember here is that HIV, it's a single-stranded, it's a positive sense RNA virus, it's an envelope virus, it is an icosahedral, right? And this virus actually belongs to a family of retroviridae. You have to remember this, right? It belongs to the family of a retroviridae. Why retroviridae? Because retro is talking with a backward. And I'll tell you guys about what that means in a few seconds, all right? Now, but within this, like even though it belongs to a family of retroviridae, within this retroviridae, there's a different groups, right? There's alpha, beta, gamma, lentiviridae, right? So HIV actually belongs to the family of retroviridae, has a subclasses, which is called lentiviridae, right? That's where this HIV belongs to. And what is the concept of lenti? Lenti is referring as slow, all right? Slow, because this is a virus which slowly progress and that causes AIDS, AIDS. It basically causes AIDS. What is this called? Acquired immunodeficiency syndromes, right? Remember this. HIV is an etiological agent to cause AIDS. Now, I have to make one more thing very clear is that HIV, human immunodeficiency virus, and AIDS, acquired immunodeficiency syndrome, these are not the same thing, all right? So I have to make this very clear. Because remember, look at the name itself tells you. What is HIV means? It's human immunodeficiency virus. Basically, it is going to attack our immune cells where, what are we going to have? We're going to have basically deficiency in our immune cells. And basically, whenever your immune cells actually, specifically T cells, when it falls below 200, we call them AIDS, acquired immunodeficiency syndromes. Now, it says acquired, meaning that this is not a congenital. This is not a congenital, right? You basically, throughout your lifetime, basically, you acquired deficiency on your immune cells that basically result into, or that basically give you these AIDS, acquired immunodeficiency syndromes, all right? So this is the concept that I want you guys to remember, all right? Now, before we go and talk about it, let's talk a little bit about this, the history of this whole concept of the HIV. Let me just give you, not going to go super detailed about it, but let me just give you an idea. This HIV was first discovered in 1981 in US, all right? In 1981 in USA, what happens is that, they actually, in US, what happens is that they found that there is this immune cell sort of conditions that has, there's deficient immune cells and sort of like a pneumocystic, this opportunity infections, induce sort of a, symptom that was actually seen in homosexual male, right, in 1981, right? And that's where the first is actually, discovered this. And initially, what happens is that this HIV was not called, it wasn't this name. It was a different name that was that time given, right, in 1980s. Usually it used to call this as lymph. It's called lymph adenopathy, right? Lymph adenopathy associated virus, all right? And later it changed and, they called that as HIV. And even during the 1980s, just a fun fact, even the AIDS, there was a different names was given for this. They used to call this as a gay-related immune disorder. But remember, but it doesn't only infect, let's say, the homosexual males, but it infects like anyone too. So that's the reason why, the name has been changed, and now we actually call this AIDS, right? AIDS, this acquired immunodeficiency syndrome. Now, a little bit something about this, what is the whole concept of this? How did we actually get this HIV virus, right? How did we get it? And there has been different theories has been written down there, but one of the hypotheses is that, there is something called monkey. What is monkey? This is an African green monkey, right? And what happens is that something called African, which is called simian, all right? The simian, basically simian monkey, that's the name of the monkey, it contains something called simian immunodeficiency, let's say, the virus, right? So, there has been hypothesis that, somehow that we were maybe the people in Africa, they got infected from this, the monkey, or they were somehow, they ate the meat, or maybe there was a, maybe scientists may have used, let's say, this monkey to actually make a polio vaccines against it, right? So through that we human perhaps probably, got infected. But these are just a theory, right? I mean, I don't know for a fact that whether or not that is the case, but I just wanted to quickly touch on this, all right? So this is a little bit of history that I wanted to address it to you guys, all right? Now, after this, let's talk about this. Let's talk a little bit of the epidemiology, all right? Epidemiology. And what is the whole concept of epidemiology is that since the 1980s, right, about 75 million people have been infected by HIV. And out of that, more than 30 million people have died since the epidemic that happened since 1980, right? There's a huge amount of people that have died over the years. And currently, based on WHO, currently about 38 million people have been infected by HIV. Okay, this was a report from 2019, all right? 38 million people, right? And they're currently living with HIV globally, all right? Now, and especially if you want to remember, if you want to think about US, USA usually gets every year, let's just say, gets about 36,000 new cases. All right. So this is a huge amount of people who actually has got this virus, and as we know that there is no cure for this virus. As you get the virus, it's going to stay on your body for the rest of your life, right? And so that's why, we need to understand the why we're not able to find the cure of it, right? And what is the whole, the concept of how HIV gets in our systems, and how it's infect the human beings, and how how the virus replication occurs. We'll talk about it here, all right? Now, after this, a little bit of epidemiology. Let's talk about how this virus, this HIV, get transmission. So we'll talk about here whole concept of the transmissions of the HIV. Okay. Remember, there is the transmissions of the HIV, it depends, usually it depends on the viral load, the amount of the variant, the HIV virus particles that are present, in your what? In your fluid. Like for example, what are those fluid? Let's write it down here. The very, look, high content, number one, obviously your blood, all right? Blood. Second, all right, that is seminal fluid, or let's say pre-seminal fluid, all right, before the ejaculations. Or let's just say that not only this, like vaginal, let's just say vaginal secretions, all right, or even your like fluid, like for example your body cavity fluid, like for example, your peritoneal cavity, or let's say that your pericardial cavity, or let's say pleural cavity, all those cavities, all right? Not only this, what about the lymph fluid, right? Or not only there, or even cerebrospinal fluid, right? Look at this. This is the area, right? These are the places where you can find the highest concentrations of the HIV, right? Not only there, like look, even the breastfeeding, like for example, when mother gives a breastfeeding to the children, it's in there, right? Like basically in the breast milk, right? So this is where you can find the highest concentrations of the viral, the HIV viral load there, right? So basically, basically through this, like anyhow, if you have a directly, let's just say that, if you're able to, for example, now we're talking about the mode of transmissions, right? There were the heavily concentrated amount of viral load is there, right? So, but how the heck this get transmitted to someone else, right? So let's think about if the infected HIV person is there, right? One of the things is like, you see, what is this I do? Here, this is basically your uterus, right? And this is your vagina, right? So whenever you do let's say the coitus, like, whenever you have the physical, right? Sexual contact with, let's just say, the infected person, like vaginal sex, that is the one way, like one of the ways, like a vaginal sex. The mode of transmission is vaginal sex, right? But more than that is let's write down here, actually number one, this one, anal sex. Okay, anal sex has the highest rate of transmission. One of the reasons is because, remember, what happens is that if infected persons is able to, somehow get contact into the mucous membranes, right, then it can, or through the blood, the virus can actually directly, it can get into the persons, right? So, and the anal sex, because why? Remember, the area down there is extremely delicate, okay? So because of that, sometimes during the sex, what happens is that, some of the mucosa might get ruptured, and you may get contact with there's a secretions is there, like this, the seminal secretion is there. On top of that, like, it might even get contact with the blood, right? So that can actually, that's why it has the higher risk, okay, of transmissions. Same with the vaginal sex. Third, like oral sex also, right? Oral sex is very important. Or let's just say the blood, through the blood. Like sometimes, like it's commonly seen in the people who are actually, sharing needles, like IV, those, the drug users, or let's just say, healthcare setting, sometimes accidentally, right, infected, whenever you're drawing the blood of the infected HIV persons, accidentally they may, they may inject themselves. So that is the one way. But usually, that the risk of getting HIV is relatively less though, but still it is a possible for that, right? Now, not only that, like for example, sharing needles, like for example, if you're like, let's say, if you're making like tattoos and using the same needles, right? That, or let's just say if you're sharing like toothbrushes, that is also one of the way that, you can actually get contact with the mucous membrane. But I'm talking about the person who is infected with HIV. That's how we can get into, right? Obviously, seminal, like basically, with the sex, obviously, anal sex is there, the key, obviously, and then, and not only this, like basically, so the way of transmission is obviously one is through the basically vaginal or anal sex, right? That is one of the reasons. Other thing is obviously oral sex is there, the blood is there, and in children specifically, in children it is very important. Remember, even basically what happens is that the baby, if this is like a uterus, right? What happens if let's just say this is a baby, all right? If it's a baby, is this right? So basically, what happens is that there is a transplacenta transmissions, right? Or we also call them a vertical transmissions, right? During that, the mother, because the baby mucous members are extremely like delicate, that sensitive, they're thin, right? So that time also the baby can get contact with the mother's like those secretions, right? The fluid. So the baby can get that, right? Other is that, as I said, the breast milk, breastfeeding, right? Because HIV is present on the breast milk. So that is also one other way of actually getting, this is the mode of transmission, getting to another person's, right? So here, quickly, so there are a couple of topics we talked about, right? Highest way of transmitting to someone, if you're infected with HIV, is obviously the anal sex or vaginal sex, obviously oral sex, right? Or let's say we say blood directly getting inoculated with that, or sharing needles, obviously breastfeeding, or like let's say vertical transmissions in babies. That's how the babies can actually get the HIV virus. And remember, if the infected mother has actually HIV, there are drugs that we give, like anti-HIV therapy. And when you give the drugs, the risk of actually passing to the babies, the risk significantly drops down to maybe like 1 to like 2%, right? So that is why it is really important to like actually do screening for the mother, whether or not like she has an HIV or not. All right, so that is very, very important. On top of that, let me just also quickly talk about this like couple of myths around there, right? Like there are like some HIVs are basically like found not only there, like basically in your saliva too, right? Or let's say in tears also, even in urine also, right? Like those HIVs are found. But remember, the viral load is extremely low around here, right? So usually, right, it is not, transmissible to someone else, right? So by basically, let's say, hugging, let's just say hugging, all right? Or let's just say, kissing, hugging, kissing, or let's just say sharing utensils, or like shaking hands, right? Shaking hands. Or let's just say, or through toilet sits, let's just say, through toilet sits, or through like airdrops, right? Airdrops, these HIV do not get transmitted to other persons, right? The infected HIV person, by kissing, hugging, sharing utensils, you don't transfer to someone else. HIV, and even in the toilet sits, like, you don't, by sitting on the toilet sits of someone else who used it, it doesn't pass it to the other person, the person who was not infected. One of the reasons why is because HIV outside of the human host, because we're the host, it does not survive for that long, maybe like relatively like few minutes, but more than that, HIV do not get, they do not survive at all. So just remember this, right? So that is the reason why, I wanted to quickly mention this, that, by sharing all this, any of this, it is not, let's say, transmissible, all right? Not transmissible. Now, after this, right? After this, let's talk about this whole properties of the virus, all right? That's what we're going to talk about. If you look at this diagram that I drew, right? If I just quickly, it is a mid-sized, this HIV virus, okay? And this HIV virus has this, this black color, it is called glycoproteins 120, all right? And this is a transmembrane protein. Remember, this is surface protein, we'll write down the SU. And this transmembrane proteins right here, which is basically called glycoprotein 41, and this is a transmembrane proteins, all right? We'll just label it and we'll go very detailed about it. So this is a lipid membrane in the blue, right? The lipid membrane contains, remember, lipid membrane is actually, this is, the virus has stolen, like basically, this lipid membrane from the host cells, right? And within there are these different proteins are there, right? Glycoprotein 120, which is this black, and this glycoprotein 41 is a transmembrane protein, which is this green, okay? So okay, let me write down this glycoprotein with the black so you don't get confused on this, all right? So this is basically, this black is a glycoprotein 120, all right? Now, this is a transmembrane. Now, these are basically an enveloped proteins, all right? Remember how we said that this is an enveloped, right? Enveloped proteins, all right? There's an enveloped proteins. Now, so HIV has an enveloped proteins, and then it also has this core protein. We call them inner core proteins. Let's just write down here, okay? So inner core proteins, inner core proteins, right? And inner core proteins, it has two layers, okay? One is the matrix protein, all right? Which is also called this as a P, all right? This is P17, and it has this capsid protein, okay? This is a capsid protein. And this capsid protein is very important. It is P24. And you know why this whole thing P17, P24? What is this concept of 120, 41? These are based on, these are actually 24,000, 17,000, 120,000, 41,000 daltons. These are basically, they're given based on the molecular weight of that proteins, all right? So remember that there's an envelope proteins, right? So the envelope proteins are made by the glycoproteins 120 and glycoprotein 41, right? Then after that, what happens is inner core, right? Inner coat. Within the inner core, there are two types of proteins there. You see this one right here? This is actually called matrix proteins, all right? It is very important for stabilizing the functions of especially this glycoprotein or glycoprotein 41, right? And this is a P17, right? Now, what is the more important thing is this one. This is your P24, this one right here, right? This is P24, right? Or capsid proteins, right? Because this is what is called icosahedral. Why icosahedral? Because it has like 20 different like sort of a faces, right? So it's because of that, you know, we call them icosahedral structure. And these are capsid proteins, and this P24 is a very, very important for clinical marker because this P24 is a relatively stable molecules. It is an immunogenic, all right? What is it? It is immunogenic, all right? It is an immunogenic. But we'll talk more about this when we get there, but they were just like drawing the structures, all right? And inside this capsid, right, what you have is that you have a 2 RNA genome, right? Remember, single-stranded, right? There's a single-stranded right here. You see this? There's a single-stranded RNA, but they have a diploid, which means that they have two, right? Two, or it's a diploid RNA genome. That's what they have. But remember, they are identical to one another, but they are not complementary to one another, okay? Because they are not complementary. These are just two separate like RNA genome they have, right? And this is where they have all those enzymes, right? Like for example, you can remember this as a rip paw inside there. Let me write down here. 1, okay, you have a 2 here, you have a 3 here, okay? And what are this? You can think about this here, inside there, they have this enzyme. This is called, it's a reverse transcriptase. We'll talk about this. It has this integrase here, and it also has a protease, all right? It has this different enzymes there, okay? So these are like a general structures of your HIV virus, all right? Now, it is very important that you know the structures of the HIV. Again, let's repeat one more time. It is an envelope proteins, right? The envelope proteins are made by two different types of functional proteins, glycoprotein 120 and glycoprotein 41, right? And inside that you have an inner layer or inner core or inner coat, and that inner coat has two types of like proteins. Actually, you have a matrix protein, which is made by P17, and there's a capsid protein, which is an icosahedral that is actually made by the P24. And there is also P7 protein. P7 also protein is there, but that's not clinically relevant for now. We'll just focus more on this. All right. Now let's talk about how does this HIV, infect our cells? Like how the heck the HIV infects us, right? But before we go there, we have to talk about, there are different types of HIVs there, right? And there are two types of HIV in the family. We'll write down here. Remember, this is a positive sense single-stranded RNA virus, right? And then we'll draw here, right? It belongs to the family of Retroviridae, right? And within the family of, it belongs to the family of Lentiviridae, right? Now, in the Lentiviridae, you have two types of HIV. You have HIV-1, and you also have a HIV-2 virus. Now, what is the whole concept of HIV-1 and HIV-2 virus? And you need to remember this, all right? The HIV-1 is the one that we're going to talk about in this lecture, okay? Because HIV-1 is highly virulent, right? What is it? It's highly, let's write down here. It's highly virulent. We'll say it is highly virulent and also transmissible, all right? Transmissible comparing to the HIV-2. And second concept here is that this HIV-2 is less virulent. It's less virulent and less transmissible. And usually, this is more prevalent in West Africa, right? Even though there has been cases of the HIV-2 that are seen in not only, let's say, in West Africa, but also in some part of the Asia or even other countries like Latin America, you do see there too. But still, this is less transmissible, less virulent, and also the drugs that we have made for this one, this nucleoside, like those reverse transcriptor, non-nucleoside, one of those drugs, it does not actually work because we have designed that drug to target the HIV-1 type, okay? And remember, this is highly virulent, right? And this is also, this HIV-1 is the one that is actually, it's present everywhere. It's in the global. This is the one, it has caused the global, basically, pandemic. All right, the global pandemic has been caused by this HIV-1, and within the HIV-1, there's a different genotypes. Okay, within this HIV, let's just write down here. Within this HIV-1, right, there's a different xenotypes. We have subtyped into different genotypes, and you have M type, you have N type, you have O type, and you have a P type, right? And what is this name? What is that? They're talking about main ones, all right? The main ones. And so today we'll be talking about the HIV-1, the M type, okay? The M tropism. We'll write down tropism here too, and we'll talk about what that means too, all right? This is the one. The HIV-2 also has a different groups. Usually it's labeled from A to H, all right? Now, so this is the concept of this HIV virus, right? Now you have to remember this is the one we're going to talk about, the M type. Now, and usually that I mentioned the word of the tropisms, right? And what is this? The tropism means, right? Look, the tropism basically means this: when the HIV-1, which is the main type, when it attacks our human cells, right? It has a specific, affinity to the specific cells. It recognizes specific cells and it goes bind to that. So it has a tropism to the specific to the cells, right? That's what we're talking about. And what is that? What is that specific? What cells it goes bind into? That's what we're going to talk about. And that cells actually, let's write down here. That cells actually, this HIV virus, we'll write down here. That virus, let's just say HIV, right? This HIV-1, which is M tropism, right? let's just MT type. Let's just say we'll just MT, okay, tropism. It has specifically recognizes a receptor or cells that are CD4 positive cells. It is a very, very important concept. You need to remember this. And what are those CD4 positive cells, right? This is the one. I'm going to write it down here, right? The one is, okay, we'll just write down here. One is, look, there is the cells which is called, look, monocytes, right? Monocytes, very importantly, not only monocytes is there, let's just say this isn't, what is this? Let's just say not only this, like macrophages, all right? Dendritic cells, all right? Dendritic cells are very important, right? So monocytes, macrophages, dendritic cells, also like let's write down here, microglia, I'll write down here, microglia, which are actually found in the, it is a monocyte derivatives, but these are resident macrophages that are present on your brain, right? Those are called microglia. Those are CD4 positive cells, right? So you have to remember monocytes, macrophages, like dendritic cells, and also the naive, those T cells, right? Also some of those T regulatory cells, also CD4 positive cells, right? So these are, so and also we'll just write down the T, obviously it's very important concept, we'll just write down here, the T helper cells, right? Those are also CD4 positive cells, like T helper cells, like the T helper 1 and T helper 2, right? This is an important concept. So remember, so this is why, we say it is a tropism to basically for CD4 positive cells, right? But there's also one more concept that we will talk about it. Yeah, when initially it infects, right? Not only it attacks the CD4 positive, but it also has a co-receptors, right? And that's what we're going to talk about here. Now, what is the whole concept of this co-receptor? Let's just say that we got infected to the virus, right? The initial infections. How the heck it got into this, right? Now let's just say that we'll make a virus here, right? We'll make a virus right here, right? Now this is our virus, right? This is a, let's just say that, this is a glycoproteins, all right? Glycoproteins, let's just say this. And then I'll basically make a different structures. Let's just say that this is a nucleocapsid, and inside there, this is the red. This is RNA genome, right? And then obviously you also have this, all this black. What is this? This one is my matrix protein, this is a nucleocapsid, this is a P24, right? Now this is a little different drawing from this one, but you got the concept, right? Now this is your gut. Now let's just say that when it, like let's just say that if we got, let's just say infected by the HIV, right? Let's say if you're from the, if you're having like let's just say anal sex, then that means that it's gonna get infected from your, the mucosa down there, right? And the mucosa associated lymphoid tissues are located there, right? MALT is very important. The MALT, M-A-L-T, mucosa associated lymphoid tissue, because they contain a lot of these lymphoid cells, right? Like for example, they contain a lot of these dendritic cells, the macrophages, there's all these T helper cells out there, right? And usually, like, if you're talking about, let's just say that, through the blood, if it got it, it can usually get from this, the GI system, right? And it can, get infected, those GERD, basically what? Gastric associated lymphoid tissue, right? Usually it can, and from the MALT, it can actually get, or it can directly attack the, this, gastric associated lymphoid tissues as well. And usually because the reason why, they target these areas is because there's a lot of these lymphoid cells are present there. The CD4 positive cells, majority are present there, right? So usually initial infection from the colonic mucosa, right? From the mucosa, like, it gets infect the cells, right? Now that's what happens. Now let's think about it. Let's talk about how that it gets infected, right? One of the most important things you guys remember is that initial infections is, when, let's say, when the virus gets into your body, right? Or in your mucosa, it does not infect the T cells. You have to remember, it actually, usually it infects like macrophages, right? Macrophages, why? Because of these reasons here. Let me erase this, right? The reason why is because if I make this, let's just say, okay, we'll just make this as a whole concept of this macrophages, right? This is a macrophages, or we'll say this is a macrophage, okay, for our case. Now, what happens is that this macrophage, they show basically a receptor right here, okay? And remember, this is receptor, this is basically your CD4 positive, right? Now, and this basically, this guy right here, I'll erase this, right? I'll draw in green, okay? But let's just say this is green actually, and then I'll make this as actually in black. I'll make this in black. This, all right? What happens? What happens is that this is CD4. It can actually go and bind into this. And the glycoprotein, this black, which get attached. It's a glycoprotein 120. Remember, glycoprotein 120 is for the attachment and binding. Okay, let's just write down here. Attachment or binding, like attachment plus binding has to do with what? Glycoprotein 120. And you have to remember this because there's a drugs works there, right? So glycoprotein 120, and then see, like basically look, it binds to this CD4 positive cells, right? But when it goes back, what happens is that, this CD4 positive cells go conformational changes, right? What happens? And what happens is that basically result into, what happens is that basically expressions of this another cells right here, right? Another receptor actually, and this receptor you have to remember. Actually, you know what? This receptor is Y. It looks like this. Why? It kind of looks like a C. Why am I drawing C? This C is referring as C, okay, CCR5, or also called them a CD195. What is this? Remember, this CCR5 is a co-receptor. What does this mean? But this CC, which means that this is actually chemokines. The C referring as a chemokines. Remember, you have to remember this. Chemokines receptor 5 or clusters of differences 195, right? This is what you have to remember. And the macrophages, all right, and the dendritic cells or monocytes or microglia, they show this CCR5. It's because of that, this virus, the initial virus, it has a tropism for basically the CCR5. That's what we're talking about. Later what happens is that, this virus goes through a lot of mutations, and basically what happens is that they can go and bind into another type of co-receptor, which is basically called, let me write down here. Okay, we'll just make this another type, which is called CXCR, basically 4, or we also call them a CD184. Remember this. This is for the late infections, all right? Remember, initially, what happens, it binds to the CD4 positive cells. CD4 positive cells go conformation changes, right? And really what happens is that then glycoprotein 4, this guy right here, right? It basically what happens, it goes and bind, basically let's just say this goes and bind into this. See what I mean? It goes and bind into this, the CCR5, right? And basically, when it goes bind into that basically gives a firm attachment, right? That's what it happens. But this has happened, the initial infection to the cells, to the macrophages or dendritic cells, it is by the CCR5 again. And later what happens, because because this virus prone to go a lot of this mutations and all, right? Later what happens is that they will show tropisms for CCR5, right? That's what happens. The later that happens, right? So this is something that I want you to remember this, right? So late is CXCR4, all right? Or we also call them a chemokines receptor 4 or clusters of 1 differentiations. And these are used, and these receptors are present on your T cells, okay? Your T helper cells, right? The activated T helper cells present this. That is why, right? Remember the name of the virus. It's a human immunodeficiency virus. The virus, which once started hijacking the T cells, with the CXCR4, what happens is that the viral, the immune cells, the T helper cells, they're going to start decreasing, right? That's what's going to happen, right? And that basically slowly progresses to the AIDS. But remember, AIDS, one more thing, AIDS does not happen like, 1 day or 2 weeks or 3 months or 4 years, right? The incubation period for the AIDS or to get AIDS-like syndrome, it's about, like like 6 to 10 years, right? That is usually the incubation period. And that is also, depends on, let's say the person is on the drugs or not, all right? The drugs or not, that's very important, that you have to remember this, all right? Now, then after that, let's just say that initially infected, right? The mucosal area, mucosal lymph tissues and all, right? And through the macrophages or the dendritic cells, what happens is they hijack the cells, right? That's what happens. They got into the cells. That happens with this co-receptor. Now, one of the things that happens is that once it get hijacked, right? Now what happens is this macrophages, right? Because remember macrophages, dendritic, these are these are antigen presenting cells, right? They can actually bring, and then they actually bring it to what? They can actually bring, and then they can basically, bring this particles, and they can actually present to this, like all this, like, what is this called? T cells, right? They can actually present T cells, and T cells can respond to that, right? So by doing that, you can get lymphadenopathy. One of the commonly symptoms that initially face, you can even get generalized lymphadenopathy, right? That's one of the things that can do, all right? Now, other thing they can do is that, when this virus can actually hijack these cells, and you can use this cell to actually make a new HIV particles. They can also do that. All right, and that is pretty dangerous when they do that. Like, now, how do they do that? Let's talk about this. Now, let's talk about how the HIV gets into the cells, and then how it basically, do hold this replication process, right? Because that is the concept that we have to talk about here. All right, now, let's erase this, and we'll talk about this. All right, now, we'll say this, that, let's say we'll use one example of, let's say that, T cells or something, right? What happens is that, let's say that, it's one of those cells like macrophages, or let's just say dendritic cells or T cells. We'll use one example, and this applies for all those cells, all right? The CD4 positive cells. Like, what happens is that, and we'll make this as let's just say this is the cell, okay? So what happens here, look, initially what happens is this, it has this, let's just say that this cells is what? Has a receptor, right? These are, what is this? These are CD4 positive cells, right? And it has a receptor, let's just say, this could be, CCR5, or we also have, let's just say for our case, we can even say CXCR4, right? I'll just write it down here. So what happens is that when the virus comes, and let's just say this binds into this, right? This is your glycoprotein 120, right? Remember, glycoprotein 120 is for the attachment and binding, right? That induces conformational changes, and that, after that, what happens is that basically result into binding into what? CCR5, right? When that happens, you see this green molecule, they have this, what is this called? Glycoprotein 41, right? This is the one is for fusion. This is also called them fusion proteins. It basically, what happens, look, it binds, the glycoprotein 120, it bound into, let's say, the CD4 positive cells, right? That causes conformational changes, and that basically induces, like, this expressions of the CCR5, basically what happens, that also binds into this glycoprotein 120, right? And when that happens, they're what? They're extremely like adhere to one another. There's a strong affinity, strong binding is there, right? And and when that happens, this glycoprotein 41, which is a transmembrane protein, right? It kind of like, it penetrates, it fuses, right? When it fuses together, what happens is that, look, it basically fuses together, and basically what really happens is that, okay, let me erase this. Okay, let me erase, you got the point, right? It basically what happens is that, when they do, it basically result into, look, it happens, it basically forms this whole like a hairpin loop kind of thing. Hairpin loop like a structure, right? It forms a hairpin loop like a structure. And then this virus, it comes inside here, right? Now you basically have this outside protein, right? Let's just say that, this glycoprotein, this, okay, we'll just say, actually, we'll just say this, guys, this transmembrane protein, right? Which is fuses with, let's say that, which is fuses with this guy, look, which is fuses with this one. When it fuses, it forms this hairpin loop, right? Hairpin loop. And this guys comes in. Now this part comes in. What comes inside here is that you have this black particles, right? What is this called? Your matrix proteins, and you have the capsid, right? You have a capsid, and inside the capsid you have two identical genome. This happens. Two identical genome with the reverse transcriptase enzymes and all that, right? That's this here. And usually, in these cells, this is like, this is one of our CD4 positive cells, right? Inside these cells, we have this is our human cells, right? We have enzymes, a proteases enzyme, which actually can breaks down this capsid. Then this capsid cap. And basically they release two particles. They release what? When you have breakdowns of all this kind of stuff, right? They basically release two identical, this RNA, right? This is RNA genome. This is very important because the two identical RNA genomes, that's what there is. And remember, because this CD4 are positive cells, right? What do they have? We'll draw it down here. They have this, our genome. We'll write down with the green. This is our DNA inside the nucleus. This is the nucleus, okay? So now, when this happens, remember how this virus inside the nucleic capsid, it has enzyme, right? What is the enzyme it brought with it? Remember we said the reverse transcriptase, right? There's a reverse transcriptase, is there, right? It also has another enzyme called integrase, right? Integrase, and also another enzyme called protease, right? It has all of them. And we'll talk about all of them. And you can remember this as a RIP, Paul, R-I-P, RIP and Paul, all right? This is important. Reverse transcriptase, integrase, and protease. And this P is referring as Paul gene. All right, we'll talk about that means. Now, what is the concept of here? The two identical RNA genomes, which are actually in the cytoplasm of our cells, our host cells, right? Now what is going to happen is that this reverse transcriptase is also called RNA-dependent DNA polymers. What is it called? It's called RNA-dependent, all right? RNA-dependent, all right? It is an RNA-dependent DNA polymers enzymes, all right? It's RNA. So what it's going to do is that I'm going to take one RNA molecules here and I'm going to draw it here, all right? So you can just remember this. I'm going to do it one here. Look, this is one RNA, right? What happens is that, it's going to start synthesizing this reverse transcriptase. It's going to start, making what? It's basically started making a complementary of this one another. That's what it's going to. And it's going to start adding the complementary to this RNA genome. Okay, this is what it's going to make. This is what's going to happen. Now, when they do this, right? And how do they get the nucleotide? They use our nucleotide that's actually present in our cells, in our cytoplasm, and they basically make the anti-complementary of this RNA. That's what this reverse transcriptase is doing. So it's basically flipping our law of this whole concept of central dogma rule, right? Remember how the central dogma rule works? Let's just correctly talk about it here. Okay, I'll just prepare here. You basically, you make DNA and we make RNA, right? From the RNA, what do you make? You make, messenger RNA, right? And you make what? Protein. This is how you make, right? So usually the DNA to RNA, this is called transcription process. And then you make translations, right? This is a central dogma, right? And then what happens, and if you DNA do replications, this is called replications, right? But remember, from the RNA, we're going backward, right? Backward, we're making DNA. That's what we're trying to do. So by doing this, and this is the function, it means you're doing reverse, right? You're doing reverse. That's why we call them this a retroviridae, right? The family of retrovirus. That's what's happening, right? So basically, it uses our nucleotide, right? Nucleotide, and then basically makes the complementary of this RNA genome. That's what it does. And that's why it's called RNA-dependent DNA polymers. That's right, remember? It is basically, it's reading the RNA. Usually DNA polymers reads what? DNA, right? DNA polymers reads basically 3 to 5 prime and synthesize 5 to 3. That's what it does, right? In this case, what is happening is it is reading the RNA and making anti-complementary proteins of that RNA. That's what it makes. That's what it does. And once you make this one, right? Now, once you make it, what happens is that second step, what they do is that, they use this template, this DNA, this is called newly made this DNA of using our nucleotide, and then what happens they synthesize, they read, and the reverse transcriptase again reads this RNA, right? This complementary, right? This DNA, and makes what? It basically makes this guy here, okay, let me draw a different one. It basically makes this structure right here. What is the structure? It makes a DNA. This is what it does. So basically, look, it makes a complementary, right? This RNA and the complementary one. Then after what happens, it uses this template to synthesize single-stranded DNA. But this single-stranded DNA is what? It's a viral DNA. It's a viral DNA. It's a single-stranded, it's a viral DNA. That's what it made, right? Now, then after that, what it did is that, then after the second thing, what happens is that, now it uses this second, right? This is a DNA molecule, right? Then what happens is that DNA molecules, now it's going to follow. Remember, this whole concept is the reverse transcriptor. What it is that it read the RNA and make complementary one another. Then after that, right? Remember, after that, what happens? It uses this DNA, right, to synthesize, like basically single-stranded DNA, and this reverse transcriptor, it basically clips and removes this RNA genome. That's what it does, right? So now, this newly made single-stranded DNA, what happens is that it go process, or they make replications of that, and you basically make what? Double-stranded. It basically makes this double-stranded DNA. That's what it makes, right? So basically, you basically have 5' to 3' and 3' to 5'. That's what it does. This is the whole concept of this reverse transcription. That's why we call them RNA-dependent DNA polymers, right? So now this is a viral HIV DNA. Okay, this is a viral DNA, or we also call them a cDNA, right? The intermediate. Now after this, what happens is that what happens is that what is the role of this viral DNA, right? Once they synthesize this viral DNA, what happens is this viral DNA, remember this viral DNA can actually get inserted into this our cell nucleus, right? Because they have a nucleus pore and it can get into there. And once it does that, because if you think about, this is our, let's just say, this is our gene, right? I mean, our DNA. What it does is that, it basically, the integrase, the enzymes, remember that the virus also have an enzyme called integrase? That enzyme integrase comes, right? And you know what integrase does? Integrase, let's just say this is I as integrase, right? This I has a two, look, it basically, look, it cuts, okay, we'll make a red. It cuts the DNA particles, okay? When it cuts this DNA particle, let's make this a little big. It cuts the DNA particles, right, or makes a nick out of it, and then it gives what? And basically what happens is that, now let's just say that your viral DNA, on the black, you have, this is a viral one, right? The viral DNA is there. It gets a nick, and it inserts this viral DNA in our DNA, right? That's what it does. So when it creates a nick, it makes this look something like this. Look, you have this, and you have something like this, right? Now, when you have this, it cut the integrase, made it like a snip, or it made a nick or cut that, our DNA, and then after what happens, it inserts this, basically what? The viral, this is viral DNA. So one of the functions of the integrase is what? Basically incorporate that the viral HIV DNA into our host cell DNA. That's what one of the things it does, right? And when it does, what is the other things happens? So when the viral DNA happens, now this is incorporated inside the cells. Now it's incorporated inside the DNA. Once it's incorporated, it's always going to be incorporated, right? That's what happens. So that's why it is very important to understand that there are different drugs that actually works on this reverse transcriptase enzymes, right? We have basically this drugs of like NNR, non-nuclear reverse transcriptor inhibitors. We have that, right? We also have a drugs that is actually nucleoside reverse transcriptase inhibitors. Those are drugs. And where do they work? Look, remember how this RNA, in order to make a new DNA, right? We need a reverse transcriptase, right? Reverse transcriptase, we need that because it is important for adding a different nucleotide sequence, right? That is one of the functions of the RNA-dependent DNA polymers or reverse transcriptase, right? So if you give a drug that actually, instead of, adding, let's say, the different nucleotides, if we basically block on adding different nucleotide, we could able to inhibit the role of RNA-dependent DNA polymers, right? And those are the drugs that are called NRTI. And one of the drugs that I probably, zidovudine or something, right? Like, let's write down here, zidovudine, right? And this is a other type of drug which basically directly and go and inhibit or block the reverse transcription enzymes. That's one of them. And we'll talk more of that in pharmacology, all right? Now, and we have drugs that actually can, basically, block or inhibit the role of the integrase enzymes, right? Basically, which doesn't allow this RNA, this basically the viral DNA to actually get incorporated into host DNA, right? Like, for example, one of the drugs is what? Dolutegravir, right? Dolutegravir, that's one of the drugs that actually work there, all right? Now the whole concept here is that once it's incorporated into this DNA, what is next? What's going to happen? But before we go over there, let's quickly review what we talked about so far. Remember, for the attachment, and then we'll write it down here, look. We'll erase this and we'll write it down here. For your attachment, right? For your attachment and your binding, right? Binding, it's your glycoprotein 120 play a role. For your fusion, right? It's a glycoprotein 41 play a role, right? The play a role, right? That's a very important concept. And then you have reverse transcriptase inhibitors, enzymes, which are very, very important for like what? Adding a new nucleotide, reading the RNA, right? The RNA genome, and making a new what? New strands of nucleotide, right? That's one of the things they did, right? Then after that, also using that to synthesize new single-stranded DNA, right? That's what this whole reverse transcriptase, or let's just say the RNA-dependent DNA polymers did, right? That's what it did. And once you made a viral DNA, or HIV, it's the role of integrase that comes and basically nicks the CD4 positive cells, the DNA, and then basically inserts the viral HIV DNA into our host cell DNA, right? That's why this is very, very dangerous, right? That's what happens. Now, one of the concepts you have to remember here is that, this RNA-dependent polymers, one is very important concept to remember, this reverse transcriptase, or let's say RNA-dependent DNA polymers, it's not like the DNA polymers that we have, right? Because basically what does that mean is that, it lacks, or it is very weakened when it comes about the proofreading activity. Meaning that whenever there is a mistakes are there, right? Usually it doesn't really fix it, right? So it is error prone. This enzyme, let's just say, this reverse transcriptase is error prone. It can make so many mutations and it doesn't fix it, right? It doesn't fix it. It's because it's extremely error prone mutations, right? It's because of what happens is that, this glycoprotein, let's say 120, all the time what happens is that it goes something called antigenic variations. You have to remember this. What does this mean? Which means that, this glycoprotein is for the attachment, right? So, if we could able to make, let's say this, let's say this, that, let's say this is black, right? In this glycoprotein, let's say 120. In this glycoprotein, let's say 120, if we could able to make, let's say, vaccine against it, right? Immunizations against it, right? To neutralize it, right? Then we could be immune to it, right? But because of this antigenic variations, they go rapid changes because this reverse transcriptase it goes mutations, right? And rapidly it goes, and because it's error prone and it makes lots of mistakes, which means it makes lots of mutations, and it is because of that, they go process of these antigenic variations, and because of what happens is that we're not able to generate vaccines so far, because of this. And it is relatively unstable proteins, right? But remember, we even said that, and that we call them an antigenic variation is because this glycoprotein 120 is for the attachment, so it is antigenic for the vaccines, but we cannot make antibody against these antigens because of the antigenic variation. Specifically, there's something called V3 looping. That is very, very important, on this glycoprotein 120, which is extremely antigenic, and that's the one that actually goes for the mutations and, the antigenic variations. And it's because of that, we're not able to make let's say, vaccines against this, right? Now, having said that, you also make antibody against, remember we talked about this glycoproteins, like P24 right here. What is glycoprotein? Right here, this, we do. This glycoprotein, which is for the capsid protein, but that is a relatively stable protein, right? But because it is inside the cell, remember it didn't play any role in like hijacking the CD4 positive cells, right? Your P24 did not participate in hijacking or infecting CD4 positive cells at all, right? It's because of that, right? It's because of that, like, even if you make antibodies against it, it is non-neutralizing. You have to remember that. Even if you make antibodies, and you do make antibody against this, but it is non-neutralizing, all right? It's not. So it's because of that, we usually use this P24 for basically the clinical marker to do the differential diagnosis, all right? If you are positive, if you have antibody against the P24, based on the ELISA test, right, then relatively more likely, you're infected with the HIV virus, all right? So we use this as for the, for your diagnosis, but it is not neutralizing because it's inside, all right? And those capsids are made by this P24, right, which is antigenic. But anyways, so you have to remember this whole concept of the glyco 124 proteins, right? Why we're not able to make, like, let's just say the vaccines against this because of the reverse transcriptase, right? Now after this, we have to talk about, what is the other concept we talk about? Let's say that now that the virus, right? Your virus is basically inside this CD4 positive cells, right? When it's inside the CD4 positive cell, what are the changes that's going to happen, right? Let's talk about that. And for that, okay, I'm gonna write it down here. Okay, it's okay. We'll draw it here. All right, it's very, very important concept here. Look, I'm gonna take this. All right, I'm gonna take this. All right, actually, let me actually erase all of this. All right, and then hopefully you remember this, because this is important concept. Look, let's just say this, let's just say that, this is, you have this cell, this whole cell, all right, is one of those CD4 positive cells, right? Now, inside there, remember how we left it, there is basically you have this DNA virus, and there's this black was what? Your viral, right? And then you have this guy, right? Now this green is our DNA, but this is the virus, like the viral particle, which is incorporated into our DNA, right? That's what happens. Now, let me take this. Remember, it has a basic coding strand, right? It has a basically coding strand, and then it has those template strand, right? That's what it has. Now, what I'm gonna do is I'm gonna draw something here, right? And you guys have to remember this, very important. Usually what happens is that, now, whenever, like, our cells, like, whenever it needs to multiply, right? Whenever it gets stimulations, right? Then it's going to replicate, right? It's going to transcribe and it's going to make a protein, right? That's what it's going to do, right? If it does not multiply, right? If it doesn't get any stimulations, right? If it did, then it basically go to the latent phase, right? Or latent infection, we can simply say that, right? But that doesn't usually happen. What happens is that, these guys, what happens, go multiply, and then, it makes a new variant particles. That's what happens, and that's a sad part. Now, let's just think about this. Here what happened here is that, the whole concept here is that, if I make this graph, right, let's just say that this is these guys have let's just make this a 5, I'm going to make this 5 to 3 prime, and actually, it has these regions, and these regions are very, very important regions, right? And I'll tell you what exactly I'm drawing in a few seconds. And then I'll make this as a green. Look. Right? Like this, let's just say this is a 1, I'm drawing this as a 1, let's just say, 5 prime to, let's just say 3 prime, right? And this is called LTR. This DNA, right? I'm taking this and I'm bringing it down here, okay? This I'm putting down here. Basically, so this is what I'm taking this, the viral DNA, and putting down here, right? They have this LTR regions, which is basically called, these are called long terminal ripids, all right? They're basically called long terminal ripids, all right? And these are 5. Now, what is the whole concept of this LTR regions, right? Long terminal ripids. This LTR regions, they contains, it's a control point for the transcriptional factor. Like for example, what are the transcription factor? Like for example, nuclear factor, kappa B, right? Or let's just say, transcription, like trans, okay, response, transacting, let's just say, response elements. And what is this whole concept of this? Or we also call them a TAR, right? So it has like those, LTR regions, long terminal ripids, right? Now, and then in here, it has a gene. This is a gene, okay? This gene is called GAG gene. Glycoaminoglycans, okay? I'm gonna write it down here actually. It is called GAG genes. It is glycoaminoglycans, did I say that? Actually, it's incorrect. GAG is referring as a group-specific antigens, all right? It is called group-specific antigens, all right? And this gene right here, which is called POL gene, referring as polymers, all right? Now we're talking about how this is going to get replications, how it's going to get transcribed, how it's going to make another HIV. That's what we're talking about now. Once it's actually inserted into our HIV, let's say our DNA, right? POL genes. And here, there's genes for that. We call them, let's just say, some of these genes are regulatory genes, or some of them are auxiliary genes, okay? And this green is for envelope gene, okay? These are different genes. Now, because we have to make this new HIV, and how actually this HIV virus makes new HIV, well, that's what we're talking about here. These GAG genes, right? Within there's basically, it's a P17 there, or P24 is there. A P7 is also there, let's just say that. Okay? And remember, this is what, and this P24, look, remember, it has, what is this? Basically, it has a protease 10, right? It has integrase, IN 32, or let's just say this reverse transcriptase, we'll say 66, right? This is how it works. It also has RNase H as well, but remember this. And you have envelope genes, which is actually encodes for glycoprotein 160. And then you have other genes right here, all right? Other genes, like for example, I'll make few of them, all right? Now maybe this one is basically maybe makes a P14. And what is P14? It is a TAT gene. Or maybe it makes another gene, which is called, let's just say, P19 gene, right? And what is a P19 gene is for? It is for RAV gene, right? Or maybe it makes another gene, which is not that, maybe it's a P23 gene, right? Or maybe, and what is P23 is referring as? It is referring as a NAF gene. And I'll explain what this means, all right? Also, there is a, not only P23 is there, right? This all, they also have the VPU gene, or let's just say, there's a, the viral ineffectivity factor is also there, right? So remember, the P14, P19, P23, the BPU and VIF also there, right? So remember this. And actually this is P27 actually NAF gene is, and then VIF is P23, right? Now what is the whole concept of this? Basically what I'm trying to say is that within this they have this long terminal ripids units, and they have each like the nuclear sequence, they encodes for like enzymes, right, or genes, right, to synthesize like all this, the functional proteins, right? So this is what it is. So what happens is that whenever, let's just say this is a DNA replication, so replications happen, right? But that DNA replication, what is going to happen? DNA replications, right? After that, what you make messenger RNA, right? And from messenger RNA, you make what? Protein, right? So we're following the central dogma rule now in this case, right? What happens? Usually what happens, whenever the cells have to synthesize, right? What happens is that, the RNA polymers too, right? The RNA, let's just say this is our RNA polymers too, right? When the RNA polymers 2 comes, it basically starts reading it, right? And then it started synthesizing what? It starts synthesizing messenger RNA, right? That's what it does. And then from the DNA replications, it uses only the template strand, right? Not the coding strand, right? It uses a template strand to actually read, right? It reads the 3 to 5 prime and synthesizes what basically, we'll just say that, and it basically, synthesizes basically the 5 to 3 prime. That's what it does, right? It basically synthesizes 5 to 3 prime. That's what it does, right? So it reads basically whole concept of this. It basically makes us like 5 to basically synthesizes, let's just say 5 to 3 prime, right? That's what it does. Now, and when it synthesizes, it basically makes a primary, let's just say primary mRNA transcript. That's what it makes, right? Remember, once it's happened that, then what happens is that, this goes basically 7-methyl guanine cap. That's where it goes, right? And this is basic, and this happens co-transcriptionally, all right? Co-transcriptional that happens, right? So so that it doesn't go degradation. And on top of that, after and remember, before the RNA polymers to finish reading it, there's another enzymes, right? Which actually puts a 7-methyl guanine cap, right, on the 5 prime end. And then once after, the RNA polymers fix reading this, right? What happens is that there's a poly-A tail additions happens, right? But this poly-A tail additions happen post-transcriptionally. Okay, this methyl cap happens co-transcriptionally, meaning that before RNA polymers done reading it, your template strands, 7-methyl guanine cap happens, right? And then after the RNA polymers done reading it, synthesizing this, the poly-A tail additions happens. And what is the concept of this? Basically, whole concept is they're trying to mature this for the maturation. So that this does not get degradation, right? That's why methyl guanine cap and poly-A tail needs to happen, all right? Now, we're not going to go super detailed about it, but let's quickly talk about now what is the next, right? Now, basically, then after that, the whole concept here is that, to enhance this property, right? To enhance the property, this nucleus, this L-terminal regions, what happens? This the long terminal regions, they have basically the nuclear factor kappa B, or this trans-acting response element, right? Or we call them a TAR. What happens is that, this protein, we talk of P14 proteins, which is called TAT proteins. These TAT proteins, right? These TAT proteins can actually go and bind into this TAR, right, receptors, and what happens? It can enhance the elongations of the RNA polymers. So RNA polymers too, right? That elongations, it's actually enhanced by this TAT proteins. That's one of the functions of this TAT protein is that, So basically, it increases, enhances the activity of the RNA polymer, so it can synthesize more new viral, right? That is one of the functions of TAT proteins. You should remember this. Sometimes they ask you this, right? Now, after this, there's also this P17. Now that after, let's just say that, the whole concept is this. You basically have you make this, then after the what? Let's just say that you synthesize. Also, just say that after that, this when you make the messenger RNA and all, what happens? This messenger RNA when you are there, let me just write down. This is okay here. Let me write on green. Green right here. This messenger RNA is there, right? And what happens is that this let's say 7.5, 3 prime, right? This is a poly-A tail is there, and there's a methyl, the 5-methyl cap guanine is there, right? All of this. So basically, what happens is that what happens? This gets basically, this gets thrown out of in cytoplasms, right? Thrown out of cytoplasms. And when you get thrown out of it, there's ribosomes, like those basically, those ribosomes, ribosomes basically, reads this, this mRNA transcript and synthesizes like different proteins. Like for example, in our case, like look, what are we trying to make? We're trying to make up those RNA genome, right? Those two here, and then you also have those, we have this nucleocapsid is there, right? We have this, all this particles is there, and then you also have like, let's just say all these different type of, let's say reverse transcriptase is there, or like ion integrase is there, protease is there. But this is aspartate protease, by the way, right? Now, they started synthesizing, like what happened? They started synthesizing all this. That's what they do, right? So when the ribosomes are reading it, this is synthesizing new proteins, right? These are all these new proteins. And you also have remember, and you also make, what is this? The glycoprotein, let's just say now, we'll just write it down here in this, you make a glycoprotein, this is a glycoprotein, let's say 160, that's what it makes, right? So it started making new synthesis. But then the whole concept is that, let's just say that, maybe like up to here, let's just say that maybe up to here, let's just say this is a nucleus, right? But after this, right, what makes it actually thrown out, this mRNA transcript into the cytoplasms? What it does is this role of the RAB protein, REV. This RAB proteins, which is basically called regulatory expression proteins, like RAB protein. And what it's RNA, it's basically RNA, right? The regulatory expression proteins, like RAB protein or viral proteins. And basically, what it does is that this P19, it actually goes and binds to this, RNA RNA, okay, response, right? It's called regulator. It basically called, it's RNA, which is regulator of viral protein, right? RNA. So it goes and binds into this, right? And basically induces this mRNA transcript to actually get thrown out in the cytoplasms. That's what this RAB protein do. Okay, the RAB protein do. Okay, so mRNA to cytoplasm, right? mRNA transcript, that is functional. TAT proteins, what is the functions of that? It basically goes and binds to TAT and enhances the RNA polymerase elongation function, right? That's what it does, the TAT protein. Increase the, like, enhancing or activity of RNA polymerase. That's what it does, right? Now, and once that, the ribosomes, like, in the rough endoplasmic reticulum, right? All this, like, what happened? All this post-translations, right? Modification, that's started going to go now, right? Then after that, what happens is that when all this endoplasmic, they'll go to the Golgi apparatus, right? We're not going to go there. They'll go to Golgi, and after that, when they go to Golgi, like, let's say all these viral particles comes in, right? And when the viral particles started coming down, what happens is that, they started coming out and they started going, and let's just say, they started going, and then what happens is that, started trying to bud off. Like, look, what happens is that whenever you have, let's just say this is a, let's just say this is a glycoprotein, 120 is there, remember, and there's also this whole, what is this called? Reverse, two parts of this reverse transcriptase enzyme. All of these enzymes are there, right? And all the STAT proteins and all of them are there. So what happens is that, when these guys are getting like assembly right here, right? Basically when they're coming together, right? What happens is that protein processing happens, right? What is called protein processing? Remember. Protein processing and assembly, right? When they're assembly, protein process basically means this role of this protease. The protease enzyme comes and it cleaves this glycoprotein 120. Right? It's not glycoprotein 120 actually. It's called glycoprotein 160 actually. This is 160. That means that I made a mistake here. The glycoprotein 160, right? So this glycoprotein 160, this comes and it cleaves glycoprotein 160, right? And it basically makes what? It makes, we'll make it the green here. It basically makes, what is this? The glycoprotein 41, and then it makes another one which is called glycoprotein 120. Remember, when you add them together, it will add to glycoprotein 161, right? But it's not that simple. But this will actually make glycoprotein 120. Okay, it's 120,000 delta molecular weight. That's what it contains. And this is an envelope gene, right? So envelope genes, right? This is an envelope gene, right? Which is actually synthesized glycoprotein 160, which gets cleaved which get cleaved by this protease enzyme. This is called protein processing, right? Protein processing and assembly is done by this basically PR, the protease enzyme, which actually cleaves the glycoprotein glycoprotein 116, and makes glycoprotein 120 and glycoprotein basically 41, right? And that is why, right? And that once it gets cleaved, a protein processing happens. Now that becomes mature virion, okay? Without the protease, this virus is not mature, okay? It is a immature form. An immature form of the virus is non-infectious, all right? That is why we have a lot of drugs that works for the protease inhibitors drugs. Like for example, protease inhibitor drugs, like for example, what are those drugs? One is, let's just say, indinavir, right? Or let's just say, lopinavir, right? We'll talk about more in pharmacology, but there are drugs which actually works and inhibit the functions of protease, inhibit the role of protein processing that is done by protease enzyme, which cleaves this basically glycoprotein 160, which is an immature form, non-infectious form, and makes more functional protein. In order to be functional, right? Even the pol gene, remember the pol gene has to get clipped down, right? The pol gene has to get clipped down into, remember this whole synthesis for pol gene, right? The pol gene has to clip down to this PRT, which is a protease stain, right? The integrase, it has to be integrase 32, right? And also the basic lower reverse transcriptase 66, right? So all of them have to be cleaved down, right? So once they get cleaved, then only they become functional, right? So that is something you have to know. So just in a bigger picture, just remember this. If you're talking about this this is basic group specific antigens. It is very important for like synthesizing. You can simply say P17 and P24. P24 is immunogenic, right? You can remember this. And whenever you talk about the pol gene, right? Pol gene, which is called polymers genes, it synthesizes 3. You can remember as RIP, RIP pol, okay? Because this RIP is referring to reverse transcriptase. This is referring as integrase, okay? And this is referring to protease, but this is aspartate protease, all right? And you have an envelope gene, okay? And these envelope genes are very, very important for doing glycoprotein, glycoprotein 41. Actually, it synthesizes glycoprotein 160, which breaks into glycoprotein 41 and glycoprotein like 120, all right? So these are the genes, like you have to remember out of your head, all right? You have to remember all of this really well. And then after that, if you could remember, like for example, some of the functions of like TAD genes, that is very important, that's extra. And also P19, right? That's what happens. Now, once it gets by protein processing, like the protease enzyme cleaves all this, it becomes like what happens is that, the functional, they started coming in as something together, and basically, they get bought off by using, let's just say this, we'll talk about it, okay? We'll try to make the same color as that one, and then, I have a green right here, right? This is basically enzymes, and you have inside there, right? Let's just say this, inside there, you have what is this? Those are basically your nucleocapsid, and you have 2 genomes, right? And you basically have the green particles right here, look. This is new virion. Okay, new HIV is made now. New HIV is made now, right? This is how usually, you synthesize the new virion, new HIV virus, right? New HIV virus. Now, quickly, what is the first thing that happens? Right? We'll quickly review this, right? Because it's very important. The first step is basically what happens is that the attachment to the glycoprotein 120. Then after the fusion happens, right? With the 41. Then after that, reverse transcriptase basically makes what? It basically takes RNA genomes and basically replicates, right? Adds a new nucleotide sequence, and those nucleotide sequence are, they got that nucleotide sequence from your cytoplasms. Then after that, basically what happens, they make single strand DNA, and then after that, the single strand DNA virus, the reverse transcriptase what? basically makes that single-stranded DNA into double-stranded DNA virus, right? Right, that's the role of reverse transcriptase. Once you have that, the integrase basically nicks or cuts the DNA, the host DNA, and basically inserts the viral, this newly made viral DNA into the host DNA, right? That's the role of integrase. That's what it does. Then after the whole concept of this, transcriptions happens, and you make, basically new proteins. And while doing that, during the protein processing and assembly, that's where the protease enzyme comes and cleaves the immature forms, right? And then basically makes the mature form of new virion particles. That's how you make a new virion particles. Now quickly we'll talk about some of that concept. Now after that, when you make this new virion particles, right, then what next? What happens to these cells, right? What is the whole concept of? And that's something you have to remember is, like how I talked about it, let's talk about the role of this NAF gene. What does this NAF means? Negative effective factor proteins. This is a P27 proteins, right? And what does this do? You have to remember this. One of the functions of this NAF protein is that, it down-regulates, okay, let's just say, this NAF protein, it down-regulates the expressions of the MHC1. That's what it does. It downregulates CD4 positive cells and also the expression of the MHC1. When the expression of MHC1 does not happen, CD8 positive cells does not recognize, does not respond to that, right? And if that happens, what happens? The cells go what? Inhibit what? They inhibit basically what? Apoptosis, right? That's what. And basically this, because of that, that causes progression to the disease or progression to the AIDS, right? That's what happens. That's the reason why it's very important. People who have this NAF protein mutation, it's significantly lower when it comes to the progression to the disease, right? So remember, so you have those other, you have other regulatory genes, like regulatory proteins, if you remember. You should remember TAT, right? RAV, and you should definitely remember NAV, okay? Negative effector protein, because it basically downregulates CD4 positive cells and all. And also, if you want to remember, you can also remember this VIR, which is viral ineffective regulatory. And what does this do? It basically inhibits the role of ApoBac, BEC, okay, BEC, ApoBac3. And by inhibiting the ApoBac3, it basically inhibits the hypermutations and degradations of the cell, right? So it's because of that, you're basically preventing the apoptosis. And why they're doing that? Because by that, what happens? They basically use this cell to what? To synthesize new virion. That's what the HIVs are doing, right? So remember, the HIV, as we talked about, there's a couple of things that HIV can do. HIV, some of the virions can actually, that can be taken by those dendritic cells, the CD, and that can actually bring it to the lymphoid, mucosa associated lymphoid tissues, or those gastric associated lymphoid tissues, right? It can bring it there, right? And by that, the CD4, those positive cells, they'll respond to that, right? Those T cells will respond to that. Or sometimes what happens, they directly hijack the cells and they use dendritic cells or macrophages to actually synthesize a new variant, a new HIV virus like this, right? And they make millions and millions of virus like this every day. And to synthesize that much of virus is enhanced by these TAT proteins, right? That's really, really important, right? That's what happens. Now, if you hijack this, let's say this T cells, right? Let's say this, then what is going to happen next then? If you hijack the T cells, what is going to happen? What is going to happen to this cell? Let's say, these T cells. Let's just write it down here. This T cells can happen two couples. Let me erase this. It can die, right? One of the reasons it can die, and how it can die is this two concept here. Let me write down here. It can die by two concepts. If it this hijack, that by what happens is that during this process, there's so many HIV particles has been made by these cells. Sometimes what happens is that there is a IFI16 is present. What is this called? It is a gamma interferon inducer, right? This is a sensor proteins, okay? Whenever there is sort of a, let's say that, some unusual activity is going on, they can sense it and they can actually, what happens? They can stimulate inflammasome reactions, right? And when what is called inflammasome? inflammatory reactions, and that can result into what? Self-destructions of the cells. But what is the self-destruction of cells? Which means that the inflammasomes, which means that, this, because this cells triggered inflammasomes, like, they produce a lot of this interleukin, like interleukin 1 beta, right? That's what they produce. And basically what happens, that results into something called pyroptosis. Like you have a let's just say pyroptosis. What does this pyroptosis means? Pyroptosis basically means what? Pyroptosis basically means that self-destructions of the cell, but due to inflammation. That's a pyroptosis. These cells also sometimes, that they do lead into activations of caspases too, like caspases like 1, for example, and that can also cause this, let's just say, apoptosis. Apoptosis can also happen into this cells. So this is how, like, the cells actually died, the T cells get died, all right? Now, so guys, like here in this class, like, we talked about, like, how, let's just say, what is HIV, a little bit of history, a little bit of epidemiology, and we also talked about the, the mode of transmissions, and we definitely talked about the, how the viral gets replications and how there's a new, the virus are produced, right? We talked about this, right? In next class, we'll talk about the graph, okay? That is, and then also we'll talk about the treatments, and obviously we'll also talk about the pharmacology, right?